Hello, everyone, and welcome to today's lecture on the membrane system. So first up, what are membranes? We need to talk about what these are in the first place. So membranes are a very thin layer of tissue that covers structures. So lots of structures, which we'll talk about. They generally line all the body cavities, and they cover things such as organs, which includes the brain and the spinal cord. More specifically, in our study for cranial, this is relevant. Membranes will have both an inner surface and an outer surface. As you can see here in the picture on the right, this would be our outer surface of the membrane, and then our inner surface of a membrane. Now, the purpose of membranes is generally to provide protection, to transport fluids between the cells themselves or between layers of membranes. They also provide lubrication, so movement of other structures upon other structures, and absorption. So they're taking part in that waste removal process as well. So in humans, some common examples that we would think of are the mucous membranes. Those are in our nose and our mouth. They're lining all the insides of that. We have them around our organs, such as stomach, heart, liver, etc. And they are going to be around the spinal cord, and that is called the dural tube, which is what our lecture is going to get into. Now, for the horses, they have all the same membranes as us because they are mammals. So let's look at the cranial specific membrane system. So in terms of the cranial membranes, there are three. These are the membranes that are going to surround the brain and the spinal cord, and collectively, they're known as the meninges. So if we look at this example on the right, we have our skin. Okay, so this is going to be on the outside of the skull. And we have our bone, that's our skull. And the first layer closest to the skull is going to be the dura mater. That's our outermost layer. Then we're going to have a middle layer called the arachnoid mater. Okay. And then we'll have an inner layer called the pia mater. We're going to discuss each of these a little more in depth, but collectively, these three layers are what is known as the meninges. So looking at the dura mater first, as mentioned, this is the outermost covering of the brain and the spinal cord, so closest to the surface. This is forming the periosteum, so that's bony portion of the skull. So the dura is actually continuous with the skull here and is actually helping to form that periosteum. Now, the dura mater is going to subdivide into septas, which separate the cavity into compartments. So what that means is that the dura mater, if we're thinking about a human skull, to give an example, so this is my skull, the dura mater is actually going to invaginate and divide into septas. Okay, so this inside would be a septa. And one of the big ones that we're going to work with in our course is the dural venous sinuses. So there's going to be a sinus that actually runs between this invagination of the septa. The dura mater is also the strongest of all the membranes and the most sensitive. So this is really important. The dura mater is very inflexible. And this is great. We want that inflexibility because it's protecting our brain and spinal cord. So it has to be pretty inflexible because that's a pretty important structure to be guarding. In contrast to our arachnoid mater is a little less flexible. Or sorry, a little more flexible, excuse me. So the arachnoid mater is the next layer down. Now this layer is a little more thin and fibrous. It's also non-vascular, so that it does not have a blood supply and it does not have sensory nerves. So this is a lot of times important because of severe injuries. If we were actually sensing all of that pain, it would be debilitating, right? So it is almost a saving grace that there are no sensory nerves at this layer. It looks like almost like a spider web under a microscope. So very interconnected weaving of the tissues. And it's going to lie between the dura and the pia. Now, the space between the arachnoid mater and the pia mater is a very important space. So this area here, this is known as the subarachnoid space, and this is where all of our cerebrospinal fluid is circulating. So it's containing all of the CSF. So that is very important for our work because a lot of the goals of the work that we're doing in cranial is to promote the flow of this cerebral spinal fluid. So it's in that subarachnoid space that we're going to have CSF circulating. Then looking at the final innermost layer is the pia mater. Now this is like the nourishment layer. It's going to be actually feeding and supplying the outside of brain with blood. So it also does not have sensory nerves, so we're not going to be picking up pain stimuli there, but it does have vasomotor nerves, meaning blood vessel innervation, because it is providing, as you can see some of the red in here, blood supply to the brain and also venous return, so taking blood away. Now this layer is continuous with the spinal cord. Okay, so it's not pictured here. It will be in images coming up, but that pia mater is actually going all the way around and it's going to be continuous with the spinal cord as it heads down and around the entire cord. The choroid plexuses, so these are the structures that are producing the CSF and they're located within this region here. Choroid plexuses are actually derived from folds within the pia mater. Choroid plexuses are very important because they are going to supply some of our ventricles with CSF. Again, this is where the fluid is being produced, but it's originating from these folds in the pia mater, and that pia mater is continuous around the entire spinal cord. Now we want to look at the three main functions of cranial membranes. So number one, as we just discussed, it's a cavity that's going to house and circulate CSF. Number two, it's going to provide very important protection to the brain from injury and disease. So it's providing a barrier to the brain. Number three, it's going to provide nutrition and blood to the brain. So remember that pia mater, the innermost layer, is going to be providing that nutrition and blood to the brain. So those are some pretty important functions, right? We definitely want cerebral spinal fluid. That's what's feeding our entire nervous system. We definitely want to protect our brain and spinal cord from injury. And we definitely want blood to be coming in and out of those tissues, and we want nutrition to be reaching them. Now we need to talk about a structure called the dural tube. So this is that continuation of the meningeal layers that is going to not only go around the entire brain, but it's going to extend and be continuous all the way around the entire length of the spinal cord. It's going to envelop the spinal cord, and therefore that's enveloping that entire CNS. So the whole nervous system is enveloped in this dural tube. Now, this is continuous, as mentioned, from head to sacrum, but it does have some firm attachment sites. So it's actually going to have some bony attachments at the base of C0, so the back of the occiput here, and on the posterior aspect of C2 and C3 in the cervical vertebrae. So they're going to have some attachment sites. We want a little bit of integrity in there, not just a tube that's free flowing within the spinal canal. We do need some attachments to give it that integrity, and those are found there, as well as at S2. So sacral vertebrae 2 is another firm attachment site. Now on dissection, I found a couple that were also having attachments at C1. So keep in mind that while most of our texts are going to refer to the attachments being C0, C2, C3, and S2, there is always room for individual variation. Now, these membranes are what are called reciprocal tension membranes. Okay, so remembering that these are a continuation of the dura mater. Recall that dura is also the layer that attaches to bones. Okay, now let's just look at these individually because we do have three reciprocal tension membranes that are sort of forming that inside membrane system inside the head. So we're going to have three main ones that we're going to learn to work with. There's the falx cerebri, and this is our sort of mohawk membrane. So I love this picture because we can see not only does it show how it's lining and forming part of that periosteum, so the inside of the skull, but it has this origin site at the ethmoid bone. It's the same in the horses, and it's going to actually be this mohawk membrane that's going through the skull and then attaching at the back of the occiput, as well as joining up with the tentorium cerebellum, which is another big membrane. So this is our tentorium that transects through the falx, okay? So these are going to join forces here and become one structure. Now the tentorium is going from back to front on a horizontal plane, so it's transecting through the cranium there. Then off of that, we're going to have the falx cerebelli, and that's going to actually be a triangular fold that's going between the hemispheres of the cerebellum, which is at the base of the skull in that hindbrain area. Now, why are they called reciprocal tension membranes? Well, because they are all continuous with each other and they're joining up with each other. And if we remember back to our lecture on fascia and the concepts around tensegrity, this is the same idea here. So what it's saying, or what I'm saying, is that all of the membranes, because they are continuous and they're joining with each other, that when we have tension to one side of the cranium, it's going to create an equal and opposite pull on the other side. These membranes, though, are very good at balancing that tension and compression, which is why if we do hit our head, the membranes are able to adapt and take up some of the tension, right? So that we don't end up having our entire brain being pulled off to one side for very long, at least. So these membranes are constantly dynamically shifting and adapting to the changing pressures and tensions that are around the brain. This is another reason why when there's been severe head trauma where the brain has been impacted and we get swelling in the brain, these membranes are able to adapt and allow that swelling so that we don't actually implode inside, right? So they play a really important role. And the fact that they are dynamic and shiftable is great, but it's not great when we get fixations or long-term compressive forces in there. They are under constant tension even at rest, and this is what's maintaining the balance and integrity within the craniums. So there's always going to be a little bit of tension in the system there to keep everything in place. But then again, like I said, with impacts and injuries, they're able to shift and dynamically move to accommodate changes. Now, the dura maintains the structural integrity of the bony cranium because it's attaching right onto those bones. So it's really helping. We can see this is all dura. The falx cerebelli, cerebri, and the tentorium are dural membranes. And they're attaching on and inserting to the inside of the skull. So they're actually providing some structural integrity in there. And that's why movement of the brain and the CSF can be translated into the membranes with dynamic shifting of tension because they're actually holding these attachments. Now, if there's an impact to the cranium, as I said, the dural membranes may become twisted or compressed. Okay, so we can get shifts like so or like so. Now, often these membranes, especially when you learn more and you work more in this way, craniosacrally, we'll find that the membranes are the primary seat of some of the cranial dysfunctions that we're going to encounter because they are so strong, right? Remember, the dura is very inflexible, that they can actively distort the bones that they're actually supporting. So this is why sometimes when we see humans or horses, we'll see some severe asymmetries in the bones, and then we can do some work and we'll see that the bones shift. It's not actually that we've mobilized the cranial bones, because those are suture joints that are not so mobile like a hinge joint, but it's that the membrane tension has been removed, so the bones are going to actually appear to have shifted. Now I want to look at them sort of individually, and I've given you both a human and an equine sort of visual example. Now these are very hard to find any dissections on. So what I've done is just drawn in for you. I know it's kind of basic, but I've drawn in where these membranes are sort of attaching. So this is our side view in the human. You can see that it's going front to back, that mohawk falx cerebri. In the equine brain, it would be roughly like so, if the horse's ears are going to be up here and our eyes would be here. It's the same thing. It's going to be a mohawk attaching from their ethmoid all the way to the back of the occiput. The upper edge is attaching to the frontal, parietal, and occipital bones, so up here. The lower edge is going to lie between the two hemispheres of the brain. So it's actually invaginating and separating the two hemispheres of the brain. And it's going to attach anteriorly or cranially to the crista galli, which is located on the ethmoid bone here and here. And then for the tentorium cerebelli, that's the one that's transecting through the brain. So in the humans, if we're looking from the side, this would be our tentorium here. Same idea for the equines, just that their head is on a little bit different plane of motion than ours, but it's going to be transecting through. So I can show you on my horse cranium here. This is our equine head, okay? We would have our tentorium coming through this way on this horizontal plane, and our falx cerebri, mohawk, coming on this plane. Okay, the tentorium cerebelli lies between the cerebrum and the cerebellum. And this is especially important for horses, as the cerebellum is their control center for balance, proprioception, and movement. So some pretty critical functions occurring from the cerebellum of the horse. As we know, horses are a prey animal, and movement and balance are among the most sort of important responses that a horse may have. So when we get dysfunction through the cranium or perhaps within the membranes, we can easily see how balance and proprioception may be affected, right? Also, the tentorium is attaching to the sphenoid, which is in the center of the cranium, the temporals, which is on the side, the parietals, and the occiput. So we'll learn more about that when you do the individual bones, but for now, understanding that the tentorium is attaching to numerous cranial bones that we're learning to work with. And then lastly, we have our falx cerebelli. That's the little triangular fold that's going to go between the hemispheres of the cerebellum. Again, that will be located here. So our falx cerebelli is going to be triangular folds in this direction, our tentorium in this direction, and our falx cerebri in this direction. Now the base of the falx cerebelli is attached to the tentorium, as we said, it's inserting with that tentorium, and the apex is attached to the foramen magnum, which is the hole where the spinal cord exits, and that's on the occiput. I made a note again, this image is meant to demonstrate a cross-sectional view of the membrane that lies between the lobes of the cerebellum. It is not an exact depiction. It's my best shot at trying to give you a visual of if we're looking cross-sectionally, this would be our direction of our falx cerebelli. Now I've also included this, just a human horse comparison. I love the human one because if you actually look next to the red lines I've inserted here, that is actually the dural membrane, falx cerebri there. And then there's our tentorium, and then our falx cerebelli between the hemispheres of the cerebellum. So the human one is quite a nice visual showing the dura all around, but I inserted the lines to give you an idea about how those create septas and venous sinuses as well, because they're invaginating. In the horse, this is where the direction of the membranes would be situated. So we'd have our falx cerebri, our tentorium coming across, and our falx cerebelli there. Now this video is also a really nice visual demonstration of the cranial membranes, and I put it in here because it was very helpful to me when I was learning and putting all this information together that we've talked about today. So you can find that link in your lesson notes for this lesson. And then another visual comparison of our dural tube. So in humans, the dural tube is going around the entire spinal cord and is continuous around the brain. This is depicting the membranes we just talked about. And then in the horse, same idea. We're going to have a dural tube that's going all the way around the spine, attaching at S2, coming all the way back up and enveloping the brain. All right, so in summary, membranes are very important for protection, lubrication, nutrition, and fluid transport. Those are all critical functions that are needed to sustain life. And remember that the membranes are also what are housing and carrying that cerebral spinal fluid, which is our life force fluid. The cranial membranes, as a review, are the dura mater on the outermost layer, the arachnoid mater on the middle layer, and the pia mater on the innermost layer. The dural tube is that continuation of these membranes around the spinal cord and brain that is continuous. Remember that it has some firm attachment sites though at C0, C2, C3, and S2. And in some specimens, we may find attachments at C1 as well. Now, most importantly, understanding that these are reciprocal tension membranes, meaning they are reciprocally shifting and dynamically moving. Those membranes are the falx cerebri, the tentorium cerebelli, and the falx cerebelli. And together, they are constantly working to balance fluids and pressure in and around the brain and the bones that are attaching to them. Okay, so we know now that any change in tension can inherently affect all of these membranes because they are reciprocally related. All right, that's it for today, and I will see you in the next one.